Section: Introduction. In this section, we discuss how large language models, like OpenAI's GPT-4, have become essential in natural language processing. These models are trained on vast amounts of data to predict the next word in a sentence and have shown great abilities in tasks such as summarization and question answering. While these models have mainly been evaluated on general English tasks, there is a growing interest in using specialized models for biomedical research and healthcare purposes. These specialized models can help with tasks like extracting information from biomedical literature, analyzing clinical data from electronic health records, and interpreting radiology reports. Enhancing these domain-specific language models can speed up biomedical discoveries, reduce healthcare costs, and enhance patient care. Although models like GPT-4 and MedPom-2 have set high standards in question answering and information extraction, they come with drawbacks. These models are expensive to train and use, with computing costs increasing significantly over the years. Organizations often have to pay high fees to access these models through APIs, leading to concerns about data privacy and security. The closed nature of these models also poses challenges, as organizations become dependent on the tech companies behind the models. If the company stops supporting the model, services built on top of it could be disrupted. Additionally, the lack of transparency around the training data used for these models raises concerns about data privacy, model reliability, and the inability to fine-tune the models for specific tasks. To address these issues, we propose the development of smaller domain-specific models, like BioMedLM, which is a 2.7 billion parameter biomedical language model trained on PubMed data. BioMedLM is designed to achieve strong performance in biomedical tasks while being more accessible and transparent compared to larger models. It has shown competitive results in biomedical question answering tasks and outperformed general English models in various evaluations. BioMedLM's smaller size allows for easier fine-tuning and inference on standard hardware, ensuring data privacy and control for organizations. Its open nature and well-documented training data provide transparency and flexibility for practitioners and researchers to further enhance the model for specific tasks. By releasing BioMedLM to the public, we aim to contribute to the advancement of domain-specific language models in biomedical NLP tasks and promote transparency and accessibility in AI research. Section Summary. In this section, we discuss the dominance of large language models like GPT-4 in natural language processing, particularly in biomedical research and healthcare applications. While these models offer impressive capabilities, their high costs, closed nature, and lack of transparency pose significant challenges for organizations with limited resources and strict privacy requirements. To address these issues, we introduce BioMedLM, a smaller 2.7 billion parameter biomedical language model trained on PubMed data, which demonstrates strong performance on various biomedical tasks and aims to provide a more accessible and transparent alternative for the community. Section: Related work. In this section, we discuss two main types of models used for biomedical question answering tasks in natural language processing. The first type includes large, general English models like MedPom-2, which have been adapted for biomedical use and excel in multiple choice and multi-sentence answering tasks. On the other hand, smaller, domain-specific models such as Dragon have shown impressive performance in tasks like multiple choice questions and text classification despite their size. One notable model is GPT-Neo 2.7b, which is a GPT-style model with 2.7 billion parameters trained on a large and diverse English corpus called the pile. The pile consists of various subcorpora, including general content from Common Crawl and specialized content from sources like PubMed, GitHub, and Free Law. This model serves as a crucial baseline for comparing with domain-specific models, allowing us to better understand how the composition of the pre-training corpus affects performance in specific tasks. Another model, PubMed-BERT, is a BERT model trained on PubMed abstracts and articles, using a tokenizer tailored for biomedical purposes. It has shown significant improvements in biomedical NLP tasks compared to similar models trained on general NLP tasks, highlighting the benefits of training exclusively on PubMed data. Models like BioLink, BERT, and Dragon, trained on PubMed as well, utilize enhanced architectures and richer data sources to achieve impressive performance in biomedical question-answering tasks. BioLink BERT leverages the link structure within PubMed, while Dragon incorporates information from common biomedical knowledge graphs. Despite having fewer parameters, these models can compete with larger models due to their high-quality, domain-specific data. Galactica, a 120 billion parameter language model trained on scientific text, has demonstrated remarkable performance on scientific tasks, including biomedical question-answering tasks like PubMed QA. Similarly, BioGPT, a GPT-style model trained on PubMed, has shown state-of-the-art results on PubMed QA and other biomedical NLP tasks, emphasizing its capabilities in question-answering tasks. Furthermore, models like FLON-POM and MedPOM, fine-tuned versions of the 500 billion parameter POM language model, have set high standards in biomedical question-answering tasks. ChatGPT and GPT-4 have also shown promising results in passing medical exams without specific biomedical training, showcasing the potential of large-scale language models in general tasks. The emergence of fully open language models and datasets, promoted by the open-source community, emphasizes transparency in model and dataset construction. Models like GPT-J and GPT-Neox-20B, trained on openly available data, have paved the way for more accessible and transparent model development. In terms of model design and training, BioMedLM is an autoregressive, decoder-only transformer with 2.7 billion parameters, similar to GPT-2 in architecture. It utilizes learned absolute positional embeddings for each position in the sequence, enhancing its performance in biomedical question-answering tasks. Section Summary. In this section, we explore two main types of models used in biomedical question-answering NLP tasks: large, general English models adapted for the biomedical domain like MedPOM2, and small, domain-targeted models like Dragon. These models, such as GPT-Neo 2.7b and PubMed BERT, demonstrate the impact of pre-training corpus composition on downstream task performance and the potential of exclusively training on biomedical data for improved results. Section: BioMedLM Tokenizer. In this section, we explored how using domain-specific tokenizers, such as the one developed for BioMedLM, can significantly enhance performance in various fields like biomedicine, science, and law. By training our custom byte pair encoding (BPE) tokenizer on PubMed abstracts, we were able to represent many common biomedical terms as single tokens, unlike traditional tokenizers that split these terms into smaller subtokens. This approach ensures that important domain-specific terms are stored efficiently in embeddings, leading to improved performance in downstream tasks. We conducted experiments at the 125 million parameter scale to evaluate different design choices. The pre-training of BioMedLM involved training on a subset of the pile, specifically focusing on PubMed abstracts and full articles, totaling 34.6 billion tokens. The training process utilized flash attention to accelerate calculations and reduce memory requirements, enabling us to train the model on Mosaic ML Cloud efficiently. The training was done using Hugging Face's GPT-2 model code, Composer Training Library, and PyTorch FSDP across 128 NVIDIA A100 GPUs, completing in 6.25 days. During training, we encountered challenges with training loss divergences at smaller scales, which were resolved by switching to BF16 precision. The final training of the 2.7 billion parameter model proceeded smoothly without any divergences. Training the model up to 300 billion tokens showed steady perplexity improvements and enhanced downstream task performance, indicating the benefits of longer training despite the smaller dataset compared to other models. For fine-tuning BioMedLM for question-answering tasks like MedMCQA, MedQA, and MMLU, we employed specialized architectures tailored for multiple-choice exams. By concatenating question contexts with answer options and utilizing a linear classifier, we achieved improved accuracy in these tasks. Additionally, for tasks like PubMed QA and BioASQ, we treated questions as sequence classification examples, using a linear classifier to generate scores for different answer options. Our experiments demonstrated the effectiveness of these specialized architectures in enhancing model performance for various question-answering tasks. Section Summary. In this section, we discuss the importance of domain-specific tokenizers like the Custom Byte Pair Encoding (BPE) tokenizer used in BioMedLM, trained on PubMed abstracts, which helps preserve important terms as single tokens, improving downstream task performance. Our experiments at the 125 million parameter scale showed significant enhancements in the MedQA task when using this custom tokenizer compared to standard tokenizers, demonstrating the value of tailored tokenization for domain-specific terminology. Additionally, we fine-tuned the pre-trained BioMedLM model for question-answering tasks like MedMCQA and MedQA, showcasing improved accuracy by employing a specialized architecture for multiple-choice exams and treating questions as sequence classification examples for tasks like PubMed QA and BioASQ. Section: Optimal format for PubMed QA and BioASQ. In this section, we optimize the format for PubMed QA and BioASQ tasks. Since our BioMedLM model is unidirectional, we need to be extra careful when fine-tuning it for BioASQ and PubMed QA tasks. Unlike MedQA, where a question is followed by a passage, BioASQ and PubMed QA have a structured format with the question and context passage separated. In our initial experiments, we arranged the question at the beginning followed by the context. To assess our model's ability to generate answers, we fine-tuned it using a set of medical knowledge question-answer pairs aimed at patients. We gathered over 53,000 questions from various online sources. For example, a typical question would be, "What are the best ways to treat plantar fasciitis?" along with the corresponding answer a medical professional would provide. We then tested our model in the biomedical question-answering domain. We focused on 5 standard biomedical NLP-QA tasks: MedMCQA, MedQA, MMLU, PubMed QA, and BioASQ, as well as summarizing consumer health questions. Our model performed competitively with much larger models and often outperformed bidirectional models trained on more extensive biomedical data. Examples of these question-answering datasets can be found in the appendix. For instance, the MedMCQA dataset includes questions covering various topics from clinical to biochemistry, each with multiple choice options. The MedQA dataset presents medical scenarios with options for answers. The MMLU dataset covers a wide range of subjects at different academic levels, including some relevant to the biomedical field. The PubMed QA dataset consists of questions based on PubMed article abstracts, with yes, no, maybe answers. Similarly, the BioASQ dataset involves questions related to biomedical passages with yes, no answers. We also explored free response question answering to enable our model to provide multi-sentence answers to medical queries, akin to consulting an expert. By fine-tuning our model on health question-answer pairs from online sources, we aim to enhance its ability to generate paragraph-level responses. This training set included FAQ pages and Wikipedia subsections converted into question-answer pairs. It's crucial to fine-tune BioMedLM on such instructional data to improve its question-answering capabilities. Despite the dominance of models with billions of parameters in some medical tasks, BioMedLM achieved comparable results with larger models across various question-answering tasks. For instance, in MedMCQA, despite being much smaller, BioMedLM matched the performance of larger models due to the substantial training data. In MedQA, BioMedLM even outperformed a larger model significantly. In MMLU, BioMedLM approached the performance of larger models on medical topics. However, its performance varied across different topics, with the model excelling in some and struggling in others due to domain differences. In PubMed QA and BioASQ, BioMedLM's performance was competitive with other models, showing promise for future improvements. Section Summary. In this section, we discuss the optimal format for PubMed QA and BioASQ tasks. We highlight the importance of fine-tuning our unidirectional model, BioMedLM, for these tasks due to their structured question and context passage format. By placing the question at the beginning followed by the context, we were able to achieve competitive results in various biomedical question answering tasks, showcasing BioMedLM's effectiveness even against larger models. Section: Question Answering with Text Generation. In this section, we discuss question answering with text generation. We found that our model generally provides accurate responses to medical questions. However, there are instances where the model gives vague answers and advises consulting a medical professional. Some questions receive detailed answers, but there is a recurring issue with hallucinations, especially concerning numerical values. For example, the numerical examples in the table were incorrect, such as stating 10% instead of the actual 35% of people in the US having a vitamin D deficiency. To ensure the accuracy of numerical values in responses, additional safeguards are necessary in the application. Despite these challenges, BioMedLM shows promise as a component of such an application. We also compared BioMedLM with GPT-Neo 2.7b baseline. Through experiments fine-tuning GPT-Neo 2.7b on biomedical QA tasks, we observed that BioMedLM outperformed GPT-Neo 2.7b significantly on select tasks, including a 27% increase in accuracy on BioASQ. This highlights the potential benefits of pre-training on domain-specific tasks, as shown by the superior performance of BioMedLM. Since its release in December 2022, BioMedLM has been evaluated on various biomedical benchmarks, showcasing its effectiveness in tasks such as medical QA, clinical note section classification, text generation of medical systematic reviews, and named entity recognition. The model has also excelled in relation extraction and classification tasks across different biomedical domains, demonstrating its versatility and performance superiority in specific areas. Overall, BioMedLM's training on domain-specific knowledge from PubMed has proven beneficial for biology-related tasks, leading to improved precision and performance compared to other models.